All right, good morning, everyone. Going to switch talks now. So thank you everyone for attending today's crop clinic. My name is Dr. Adam Varenhorst, and I'll be starting our session off with a talk about our major insect pests. So if you'll bear with me just 1 second, and I'll have that up and running. All right. So as I mentioned, I'm Dr. Adam Varenhorst. I'm the SDSU field crop entomologist. And today I'm going to be speaking on major soybean insect pests. And so there are a lot we could talk about, but since we only have a limited amount of time, I'm going to keep it relatively to the point and talk about two major ones. If you have additional questions regarding other soybean insect pests, feel free to ask me those questions and I'll answer them to the best of my ability. So the first thing I have is a picture here of a soybean field that's adjacent to a cornfield, and I'm going to ask you what's wrong with this picture. So if you take a minute to look at it, you might start noticing a few things. The first thing you might notice is that, well, there seem to be some dying plants that are right up against the cornfield here. And you'll notice that there's weeds starting to emerge, so this has been an issue for a longer part of the season than just towards the end. And at first thought, everybody thinks, well, herbicide drift. Must have been a little bit hot, went right along the beans with the boom and killed some of them off. And that would make sense because if you look at the picture, this is a larger field and the dead plants go all the way along that margin between the corn and soybean. However, these crops were both tolerant to the same herbicide and it was used on both of them. So it should not be herbicide drift. And the next thoughts, well, maybe it's a disease, but if you look, it's so uniform that would be kind of unusual for a disease to follow all the way between the corn and the soybean. So the last question is then is it an insect? Well, the answer is yes. And a few years ago, this pest was really catching us off guard because like diseases, we typically don't see uniform infestations of an insect pest that follow the length of the field. So what is it? As we can see, it was actually killing the plants, and it appears to be moving from the edge of the field inwards. And this was a common occurrence for the last few years in different states, just not just South Dakota, but the trend was you'd be driving along the road or scouting your field and you'd notice that there were dead plants along the edge. And as you can see, it's not always 100% uniform death. Sometimes there are a few plants that remain, but there are a lot of dead plants along the edge of the field. And something else that we notice with a lot of these plants, even before they die, is that they become very brittle and they break and lodge very easily. And typically the break point is about 1 to 2 inches above the soil surface. And as you can see, it snaps, falls apart. These are a little bit later beans. And so you can see this lodging here, this is just caused from walking down 30-inch rows where they are canopied. We would expect you typically move those plants a little bit, but not to break them off completely. When we were seeing these plants, we noticed this discoloration right here. And we also often noticed that there was this white fungus. Now, this isn't white mold. We had it tested. It's a white saprophytic fungus, likely present due to increased sugars on the outsides of the plants. So what's causing all of this? It turned out to be these small fly larvae or maggots. These are known now as the soybean gall midge larvae. When they first were showing up, we didn't have a name for them because we hadn't seen them before. As you can see, when they're underneath the epidermis, which is where they're found, they are typically found around an area of injury. And when we first saw these in South Dakota back in 2015, we didn't see them causing any injury to the plant itself. They were feeding on a fungus that was underneath the epidermis. However, if we fast forward a few years, as we see in this picture, these insects are now actually feeding, and we can see where they're feeding under the epidermis. So things have changed, and you also notice there's no more white fungus underneath the epidermis. So these are now feeding on the plant itself. There are 3 instars of the soybean gall midge for the larva. I have them listed here. This is the first instar. These are pretty small, kind of hard to see with the naked eye, but you can still see them. They're a white, almost clear color, and they're pretty easy to tell apart from the other instars because of the fact that they're so small and light colored. The second instars are a little bit larger, and they're going to almost be a light pink, almost not any color at all. But you can see the difference here. A little bit larger, a little bit more color. And here's the third instar larva. These are the ones that are the easiest to scout for because, as you can see here, they're actually bright colored. So they're bright orange, and they're quite a bit larger than the other instars that are going to be in the plant. The adults for the soybean gall midge are about 1/4 of an inch long from the tip of the front leg to the tip of the back leg. So they're not very big. That's with the legs stretched all the way out about 1/4 of an inch. You probably won't see these out in the field unless you're either really looking or you have some traps out to collect them. One of the distinguishing characteristics about these is their legs. So if we look at the legs here, you'll notice that they have a pretty distinct pattern. It goes dark, light, dark, alternating. And so that's something that's pretty unique to these guys, as well as the fact that they have these clear wings, and then you can't see it here, but they have a bright orange abdomen. So one of the things we've learned in the last few years is that these adults are somewhat lazy, and most of the time they're resting either on the plant, or if we have cages out to trap them, they're resting on the inside of the cage. So they don't move around a ton, but they obviously move a little bit as they can infest fields. So what's a soybean gall midge infestation look like early in the season? Well, it starts out not quite as uniform as we saw in that first picture, but you'll notice here, this is a picture that was sent to me a couple years ago. First thing you'll notice probably is there's a cornfield right there. There's corn plants, and then we have this first row of soybeans, and there's some wilting and dying occurring right here. And those were checked, and they did have soybean gall midge. So you probably are starting to notice a trend here, where we have a cornfield, we start finding soybean gall midge next to it. You're not wrong. Some general scouting recommendations we've developed for soybean gall midge are you need to start looking at the second vegetative growth stage. So when you have those two trifoliates developed, at that stage, you need to be out in the field and start looking on the edges for wilting plants. Typically, it's the edge where we'll start to see these first. That's been the general trend. Another thing you want to do is look for discolored stems near the soil surface. Right in this region is where you're going to see some of that discoloration. Another thing you're going to want to look for is some swelling. Now, depending on the plant, sometimes the swelling is almost undetectable. It just varies from field to field. Sometimes it's a little bit more noticeable than other places. And another dead giveaway are the plants. If you start seeing dying plants, there's probably a soybean gall midge issue, especially when those dying plants are on the edge of the field. So I mentioned discoloration and swelling, so here are some pictures. So this is what you'll see early in the season. This is the area right here near the soil surface that you're looking for that discoloration at. And this right here is that little bit of discoloration. So typically the stem is brown right there, and we see it's a little bit darker, darker red down in. So what is this right here? It looks like a scar. Well, that's actually a scar from that first node of the cotyledons. When they drop off, we get this kind of splitting right here. And so this isn't uncommon. Researchers now, we've been looking, and we're noticing that some varieties split more, and we believe that's due to the rapid growth early in the season. So as the plants are maturing, they're growing, they're going to split the epidermis a little bit, and then that heals over. That's an issue, though, when we start thinking about soybean gall midge, and I'll talk about that a little bit more later. But that's what we're looking for. And if you also look, it's kind of hard to see. There's a little bit of swelling right here on the plant. So there's kind of this distinct line. You'll notice that it kind of curves in, and then you get a little bit of swelling. And like I said, the swelling is a little bit harder to see. Once you've observed discoloration and swelling, take your fingernail or a knife and make a slice in the epidermis and start peeling it back. When you have a soybean gall midge infestation, you're going to notice those red-orange larvae. Typically, those are the ones that pop out. You'll see even in this picture, you can see some of the other instars, but they're not quite as easy and noticeable when compared to the bright orange ones. And so, as I'll also point out, you'll not only see the larvae, in some cases, the larvae aren't on the plant anymore. We aren't sure why, but you will notice this discoloration. And it's pretty indicative that you had the soybean gall midge, even if you can't find live larvae. In some cases, I've been able to find dried up larvae in these patches. Again, I'm not sure 100% what caused that mortality. And why do we care so much about soybean gall midge? Well, we've seen a lot of yield losses associated with them in the fields that are infested. So this is a yield map from a few years ago. And as you'll see here, right on the edge, that's where the soybean gall midge infestation was the worst. And so not a lot of yield off the edge of the field. In some fields where we've taken yield, we've seen field edges that are reduced by 10 to 100%, depending on the level of infestation and how far into the field they've made it. On an average field where we see a heavy infestation, we can see anywhere from 20 to upwards of 100 feet of severe infestation with dying or dead plants. When we've looked at entire field averages, we've seen losses that range from 10 to 50%. And so you're saying, well, if the entire field had a level of infestation, how do you know? Well, we looked at previous years' field averages. So it's a rough estimate. We're still working on getting good yield data, but we definitely are seeing some losses from this pest. One of the things we've noticed, and it didn't show up great in that first picture, is even though it's just the edges of the field that look like they're decimated, we can still find soybean gall midge throughout the entire field, even in the center. And so we're starting to wonder if those center populations aren't also causing some yield loss, but maybe it's not as noticeable as where they've killed the plants. So the big issues we have with the soybean gall midge are that before 2011, there were no reports of this pest in the Midwest. In 2011, there was an article from the extension agents in Nebraska where they saw this pest in soybean. However, they were only finding it in soybean that had been hit by a hailstorm. And so they hypothesized that it was a secondary pest that took advantage of the wounds, made it into the plant, and they also noticed it feeding on a white fungus. We jump to 2015. That's when I started at SDSU. My first field call was out to a field in the southern portion of the state to look at a field where there was excessive lodging. This field had a lot of issues occurring in it. One of them, though, is that there were orange larvae present underneath the epidermis, but the larvae appeared to be feeding on a white fungus. Now, if we fast forward a couple years from that 2015 mark, that's when we started getting a lot of calls and noticed that we had a lot of issues occurring with these small orange larvae. And so they were declared a new pest of soybean. We've learned that this larva, the soybean gall midge, overwinters as larvae in the soil of infested fields. So if you remember, we typically found it next to those cornfields. That wasn't by chance. So those were previously soybean. These larvae drop out of the plant sometime towards the end of the season, and they go into the soil. They make a silken cocoon, and then they overwinter there. And they must be pretty cold hardy because then they emerge in the spring. We haven't really noticed that they die off even during some of these colder winters. In 2019, researchers determined that this is a brand new species of of gall midges. So it was previously unreported, and it's now called Riceella maxima. That's the scientific name. We'll be calling it soybean gall midge, though that's the now recognized common name for it. So that left us also scratching our heads. Here's the pest that we didn't see before 2011. And not only is it new in the Midwest, but it's new in the world. However, I should note There are only 2 people that really worked on these gall midges. Both are retired. One actually is working out of retirement, helping identify it and then helping us with some information regarding it. So some things we know for sure is that this pest emerges in the spring. We aren't sure exactly what triggers it yet. Additional issues that we might be seeing with this pest is that in all these pictures, there's multiple instars in one plant. Now, this isn't completely uncommon or unheard of with insects, because if we think about soybean aphids, you can see a lot of different sized soybean aphids on a plant. However, the difference is that soybean aphids reproduce asexually, so that's why we can see so many different stages on a single plant at a given point in time. We are under the assumption that the soybean gall midge is only emerging from eggs that are laid by the females. And so that's why it's kind of unusual that we'd see first instar in the same plant as third instar. We'd think that they'd all be more similar. Now, we don't know if this occurs yet, but there's a phenomenon or effect where gall midge larvae are known to go through what's called paidogenesis. And that's where larvae produce larvae. And that's maybe a possibility for why we can see so many different stages. We don't know yet, but that's something we are looking into. We're hoping this doesn't occur with the soybean gall midge, because that would make it a little bit more difficult to manage down the road. So for South Dakota, at this point in time, we've found this in 26 counties. The red counties are from 2018. That's where we detected it. Orange counties were 2019 detections, and these yellow ones were confirmations from 2020. You'll notice that there are still a lot of counties that aren't colored in where soybeans are grown, and it's not for not trying. We just haven't been able to find the soybean gall midge in some of these counties. So we'll continue looking. One of the reasons that we're interested in how far the distribution is that will give us an idea of where we need to start scouting more heavily. So far, one of the things we have noticed is that down in this portion, the southeast portion of the state, infestations seem to be much worse. They seem to hit a little bit earlier than they do in some of the rest of the parts of the state. And I mentioned it at the earlier part of the talk, but this isn't an issue that's unique just to South Dakota. And we're actually seeing, excuse me, soybean gall midge in 5 states. And as of 2020, there were 115 confirmed counties. So this is a pest that seems to be pretty widespread. And some of the harder hit areas, besides in the southeast portion of our state, seem to be, as you can see, between the border of Nebraska and Iowa. Now, they are in Minnesota. There are no reports yet in North Dakota. Missouri now has 2 counties. North Dakota did extensive surveying this year and did not find any. So, you know, it looks like it's probably there because it's found on the border county here, but we haven't found it yet in Roberts or Grant either. So it's probably there, and we'll just keep looking. I'll mention this a little bit more later, but if you look down here, it says found on sweet clover, and that might leave you kind of wondering what's going on, but we'll talk about that in a minute. So at the soybean gall midge, one of the things we have been doing is monitoring its emergence. We use cages. There is a soybean gall midge alert network, and I will drop that link in the chat here once my talk is done. You can sign up for this free. You will get texts and emails, and I believe a phone call also if you opt into that, when we find soybean gall midge adults, and it'll tell you where they've been found. Why might you care? Well, if you're in one of these counties that are getting hit harder, you might be interested in knowing when to go out and maybe spray adults. So that's why we're doing this early detection for the adults. For South Dakota, the first detection was the week of June 13th. You can see we had these black circles represent cages, and where you see the numbers, that's where we were seeing adults. And so cooperator with Pioneer was nice enough to put those cages out for us this year. Due to COVID, it was a little bit trickier for us to do this research. But anybody that's interested in having some cages and putting them in a field that was previously soybean, and you think you had soybean gall midge, feel free to contact me. We'd be happy to work with you. So what do we know so far? Well, there's at least 2 generations of this pest each year. Maybe there's more, we don't know yet, but we've at least seen 2 generations. We know that the eggs are laid near the base of the plant or near new wounds. These flies can't actually penetrate the soybean epidermis with their ovipositors. Something that the researchers in other states have looked at is making modified hail damage or causing hail damage to the plants using a machine. And they've seen that typically wherever there's a hailstone injury, you'll get soybean gall midge when the populations are in the area. There are increased infestations near trees and field edges. We don't know why yet. We understand why there are increased populations near the cornfields, but why near shelter belts and groves we see these increased infestations, we aren't sure yet. However, so far, there have been 2 alternative hosts recognized. They're alfalfa and sweet clover. In both cases, soybean gall midge were found on them. They were actually identified using the genetic markers to make sure that it wasn't just something that looks similar. And so the difference with soybean, though, is with alfalfa and sweet clover, they don't actually kill the plants that we've noticed so far. And as I earlier talked about, they overwinter in the soil as silken cocoons. Some other things we figured out, but we don't have a lot of evidence to support this, is it looks like later planting dates generally have lower infestation rates. So far, we have not seen a lot of evidence for variety or seed treatments making a large difference in infestations. We also haven't seen a lot of evidence for tillage. And the reason we think this is probably the issue is if you till where you're planting the soybeans, you're not going to see a big difference. And even where people have tilled their corn stalks, we still see that you can get an infestation. These are pretty deep, and so most of the time you aren't hitting them even if you're doing some tillage. We have seen some evidence that if you spray insecticides right around the time that we're seeing those adult emergences, you can reduce the infestations. Notice I say reduce, you will still get some infestations in that field. So for 2021, we're hoping to continue our monitoring of the emergence. We're building more cages to trap these things. We also want to go out and actually survey some more counties. Where we saw them this year, in the more western central portion of the state, near the river, those were areas where they actually had unusual amounts of rainfall early in the season. They were very wet. And we're starting to wonder if maybe these things don't need a little bit of extra moisture to take off in the field. We also are hoping to get some yield data this year. We weren't able to in 2020. And then we're trying to figure out a management strategy, and we're getting to a point where the kitchen sink approach, where you throw everything you have at it, might actually come into play, because in some cases this pest really does need to get under control, and in fields where it isn't, it's causing a lot of yield loss. Just a thing to add in, there's more than one gall midge you might find in a soybean field in a season. These typically show up a little bit later. They're the white mold gall midge. I bet you didn't know there could be so many gall midge larvae out in the field, but researchers in Minnesota a couple years ago came across these guys. The part that's different is, you might see it in the name here, white mold gall midge only show up in fields that have white mold infestations. And the thing with these guys is they eat the white mold. However, if you look, they can be pretty similar in appearance to the soybean gall midge. However, they're a little bit longer and lighter in color. So these are a light orange, where the other ones are almost dark orange, in some cases a pink color. So there's your big differences. But if you come across something that's in a field with white mold, it may need to get looked at a little bit closer, because it's not a home run soybean gall midge detection. It might be white mold gall midge. So in the last few minutes of my talk, I'm going to talk about soybean aphids. You guys are probably all familiar with these pear-shaped green insects that showed up, caused a lot of issues in soybean when they show up in large populations. These were first detected in 2000. The reason we care so much is if you don't spray a field that's infested, they can reduce the yield by about 40%. We use foliar insecticides to manage them. And we've used a lot more insecticides. So here's 2000 when soybean aphids showed up. This is the estimated use of the insecticides in millions of pounds. And so if we look here, this is for the Pyrethroid class insecticides. We see that in about 2004, so soybean aphids were detected in the US in 2000, built up. In 2004 to 2014, there's this dramatic increase in the amount of pyrethroids applied to soybeans. Not a surprise. Soybean aphids can really take off and cause a big headache. Why am I pointing this out? You're probably asking, well, for a pest that's been around for now moving into 21 years, that's been primarily managed using foliar insecticides. And of those insecticides, we've mainly been using Pyrethroid class active ingredients. And of the pyrethroids, we've been relying pretty heavily on lambda-cyhalothrin, the active ingredient here. Well, maybe we might have some issues with this pest now. And especially when we think that sometimes multiple applications were required to manage this pest. Our first indication that maybe we're heading towards a resistance issue is in 2012 when resistant two-spotted spider mites were detected. We spray aphids a lot. We haven't been spraying two-spotted spider mites a lot back in 2012, but all of a sudden we're seeing an issue. So maybe it's because we were spraying the soybean aphids so much. In 2015, reports of soybean aphids that were resistant or just field failures of pyrethroid applications came from Minnesota. As you can see, it wasn't a single location. It actually came from a lot of places. The big issue here is that these were retreated and still had no effect. And so they were collected, tested in the lab, and they were resistant to the pyrethroids that were being applied. Jump to 2016. Now there's still issues in Minnesota. Iowa also has an issue pop up. 2017, a lot more issues. So you can see where the soybean aphid population showed up in 2017. We tested 3 locations in South Dakota. We didn't even have large populations, but we found resistance. In 2018, I had a student actually work on this. He found populations pretty much everywhere he looked. The red indicates the 2018 resistant populations. Except we had one county here that's green. Those are actually still susceptible, so that's good news. But we did have a lot of resistant populations show up. So how do we move forward with the soybean aphid? Well, when you need to manage this pest, you have a large population, it's beyond the threshold. The first recommendation I can give you is maybe use something besides a pyrethroid class insecticide. Also, avoid those prophylactic applications where, while the insecticide's cheap, the pest might be out there. Don't do that. Please don't do that. Make sure you have the pest, make sure it's at threshold, and then apply the insecticides. And then also consider tank mixes for management. If you are dead set on using a pyrethroid, please tank mix it with something. The reason we want to try to be very careful with our management going forward is we do still have susceptible populations out in the field. We don't want to get to a point where 100% of our populations are resistant. We want to try to maintain some of those susceptible populations. It will take a long time though before, if ever, we can get back to where everything's in the population of soybean aphids susceptible, but we want to try to slow down that drive. And also, why use an insecticide that probably won't be 100% effective? So that's kind of the recommendation there. That's all I have for you guys. It looks like I'm out of time. So here's my email address if you have any questions. With COVID, I'm not in the office all of the time. So if you need something, please feel free to send me an email.
In the meantime, Adam, do you want to answer one question in the chat?
Yes, thanks, Emmanuel. I see that now. So the first question is, in your opinion, based on what we know so far, do you think adding small grain to the rotation could help reduce pressure? Douglas, I'm guessing you're talking about soybean gall midge. That's a good question. So far, we haven't seen a lot of evidence that increased rotations help. Obviously, this isn't just a you issue, though. If you have neighbors that are also growing soybean and have the issue showing up, it's kind of a vicious cycle. So even if you don't have soybeans for a year, if your neighbors did, we don't know how far this thing can move. It looks like it can move relatively far though, because they're small. They're actually kind of like soybean aphids probably in that regard. The wind is probably helping in moving these. We think shelter belts and groves are maybe actually acting as a barrier for the wind. And as a result, that's why we see increased pressure in those areas. And so even if you have a longer rotation, I can't say 100% that that's going to help you. You obviously will miss out on more infestations, but I think that when you do end up with soybean again, you still have a pretty equal chance of having a problem. Probably the best recommendations we can give people is, although it's somewhat popular, as you saw in those pictures, the split field sometimes with corn and soybean. It's probably a better idea until we get a better solution for this pest to maybe not do that. Just because if you had infested soybean, your new soybean have a very high chance of getting infested. And I probably didn't give you a great answer for that, but that's the issue with this pest is at this point in time, we don't have a lot of solutions and we have more questions than answers. Does anybody else have any questions before we transition away? Hopefully everybody was able to get the CCA credit here scanned. If there are no further questions, I will stop sharing and hand things over to Dr. Emmanuel Byamukama. He will be talking to you today about soybean fungicide resistance, SCN, which I know he likes to talk about, and also some root rot management. So Emmanuel, feel free to start sharing your presentation. It looks like you have, and I'll hand things over to you.
All right, thank you so much, Adam. Hopefully everyone can see the slides and also can hear me. So this morning, myself and Connie would like to talk a little bit about fungicide resistance, soybean cyst nematode, and one root disease that is really important in soybeans. And we thought that for us to talk about fungicide resistance, it would be nice to first look at some of the diseases that we use fungicides to manage in soybeans. And one of those is brown spot or septoria leaf spot. As you can see in these pictures, this disease can be pretty bad, especially where you have soybean on soybean. But we also noticed that this disease tends to be more severe in older or lower leaves. That's where you find this disease. These are actually two fields where I found this disease last year in soybeans. This one on the right was in Cardington County, and this one here was somewhere near Brookings. But in both fields, this was soybean on soybean, just based on the surface residue that was in this field. And there was also a lot of defoliation. So in some particular cases, brown spot can be really severe and cause yield loss. But for the majority of fields, rarely do you see this disease becoming severe. And in fact, these two fields also had narrow row spacing, so less than 20-inch row spacing is what I observed in these fields, and it was soybean on soybean. But again, for the most part, we don't see a lot of brown spot to be an issue, something that we would worry about or need to apply a fungicide. The other disease that we see in soybean is Frogeye leaf spot. This is also another disease where we might need to apply a fungicide. Excuse me. And these were also pictures taken last year. This field on the right was really bad, as you can tell, based on the severity on the leaves, and here on the left as well. So Frogeye leaf spot is another common disease that we see, especially increasingly these last 3 years. In the past, I have not seen many fields with Frogeye, but in the last 2 years especially, I've seen Frogeye really come through strongly in some fields. But again, also, it's going to be certain fields, and the main driver, it seems to me, is going to be due to the susceptibility of the variety that was planted, but also on the weather conditions. So last year, towards after soybean flowering in some parts of the state, we had good conditions for this disease to develop, and we saw it take off, although it was a little bit late in the season. So both brown spot and Frogeye are fungal pathogens that are gonna survive on residue. So they're caused by fungal pathogens that survive on the residue. And where you have high planting population, where you have that environment that's going to cause poor air movement within the canopy, you can have these diseases really develop and become major concerns. But even in water row spacing, if your variety is susceptible, you can have Frogeye reach these levels. And of course, a Frogeye leaf spot of this level is going to cause early defoliation and poor seed fill in those pods later in the season. And so when we look at fungicide, before I go to the fungicide side, let's look also at one other disease where we might use fungicides to control. So white mold, as we know, is another disease we see in soybeans. It can be pretty severe, as you can see this entire field almost having plants that are killed by white mold. Very easy to diagnose. Once you see this cotton white mycelia in the middle of the stem, that is likely to be because of, definitely that is a white mold. But if you see the top canopy, the symptoms may not be very distinct. There are other problems, like brown stem rot, can cause, also even stem canker, can cause the upper leaves to look like, they are wilting due to white mold. So the best way is to open the canopy and look at the middle of the stem. That's where you're likely to see this white mycelia. Sometimes you might see this crochet, which look like mice droppings on the stem itself, which would be a sign of the white mold pathogen. So these three main diseases in soybeans is what we usually would, that's where we would usually apply a fungicide to manage these diseases. The other foliar diseases like powdery mildew, like downy mildew, they're really not major, and so usually we don't worry about them to need to apply a fungicide. So then now looking at fungicide resistance in soybean, where are we likely to see this problem develop, where we have some fungicide resistance issues? So strobilurin fungicides, so those would be the group 11 fungicides, QI fungicides. These are going to be applied mainly to control Frogeye and brown spot diseases. So other types of fungicides like this, CDHIs and triazoles, those are usually for controlling white mold. So white mold requires specific active ingredients. So we're talking of Endura, Top Guard, Domac, Prorine. Those are specific fungicides for white mold, and those tend to have SDHI and DMI fungicides. But for other foliar diseases like Frogeye, like brown spot, one of the groups of fungicides we use for controlling these diseases is going to be strobilurins. So whether it's in a combo of formulation or solo ingredient, those are likely to be strobilurins that are used to control these other diseases. And unfortunately, this is where we're also finding resistance issues that are developing. And so as we talk more about fungicide resistance, look at the class that's being used and for controlling what disease in soybeans. So as I was mentioning, we have detected strobilurin resistance in the Cercospora sojina. This is the pathogen that causes Frogeye leaf spot in soybeans. And again, this is going to come from that fungicide that's applied to control these foliar diseases and not necessarily fungicides applied to control white mold. So this is why I want to draw a distinction that if you're applying a fungicide and you're not controlling white mold, are you applying this fungicide because you're seeing enough disease pressure to warrant the fungicide, or could you be, attracting problems, or could this fungicide that's not necessary lead to problems in the future, as we're seeing already with the Frogeye leaf spot pathogen? So as of now, we've found 28 isolates of this pathogen that causes Frogeye. to be resistant to Soybean fungicides. And we think that the resistance could be widespread in other counties. So for the 4 counties are the Southeast South Dakota counties, but in other places where Frogeye is occurring and where fungicides are used, it's possible that we could be seeing Frogeye leaf spot resistance in these other counties. So something that we need to be aware of, and especially knowing that if we're applying this fungicide, whether it's really necessary or if it could lead us into more problems in the future. So unlike what is believed out there to be the cause of fungicide resistance, applying a fungicide in itself does not cause resistance. Rather, it selects for certain fungal isolates to survive more. So in other words, if you apply the same active ingredient, you are applying a selection pressure against certain isolates, and so there's an imbalance in the nature that leads to one isolate to be predominant, and so happens that that isolate that survives the fungicide becomes dominant, and so once the fungicide is applied, it's like applying water, because the isolate that is surviving will not be affected by the fungicide. So in the nature, you have a mix of different isolates, and so happens that some may have a mutation or some change in their DNA that helps them to survive the effects of the fungicide. So when you apply a fungicide, then let's say here the blue isolates don't have the mutation to help them survive the fungicide, and so those are knocked out over time. And the red ones, because now they don't have any competition, so then they thrive better and dominate. And so that's what happens when one mode of action when one fungicide that has one mode of action is applied over and over. Over time, again, we select for that isolate that has the mechanisms to survive the fungicide. And these are many mechanisms that a fungus may have. For example, if the fungicide is interrupting the respiration process where the fungal cell makes energy, the fungus may develop a different mechanism of respiring and making energy, and hence if you apply the fungicide, the fungus can still survive. So again, here I wanted to emphasize that unlike what is commonly understood out there, that a fungicide causes resistance, a fungicide simply selects for an isolate that will survive the fungicide. So the fungicide is simply applying a selection pressure and just favoring one isolate to dominate, and all the other ones are knocked out, creating an imbalance. So again, really here the message is that fungicide we're applying it in a way that is not going to cause issues in the future, or we're applying it because it's necessary, it's warranted, and we're also applying it in a way that will not cause a resistance to develop, as I shall show you a few reminders on how to use fungicides sustainably. So looking at the different diseases that we use fungicides for, and also looking at the way fungicides can cause selection pressure, what are some of the things we can do to make sure we minimize the likelihood of a fungicide resistance from developing? So these are not new things here, just reminders that we shouldn't use the same mode of action season after season, or even more than the recommended times in a season. We need to make sure that when we're applying that fungicide, we really need it. So we restrict the number of times we're applying the fungicide. Let's apply the fungicide only when it's warranted. We have scouted there's enough disease pressure, and we need to apply a fungicide. We need to use recommended rates. So this is not very rampant in soybeans, but we've seen where low rates are used in other crops like wheat. So definitely we want to make sure that we're using a recommended rate. So again, we're not selecting against certain isolates in the field and then creating that imbalance that may lead to fungicide resistance. Of course, where possible, we want to use all the practices we have to try and control the disease and not necessarily relying on fungicides. So selecting a good variety, doing good agronomic practices, crop rotation, crop residue management where possible, to try and minimize chances of diseases developing, and therefore also eliminating the need for a fungicide. So moving on, you can help us to monitor for fungicide resistance in your operation. First of all, try to keep an eye on things. So when you apply a fungicide, don't hang up everything and then forget and go back to harvest the grain, but keep an eye on things. So go back after 2 weeks and see, did this fungicide help to control the disease, or are you still seeing symptoms develop? In fact, the first alert we had was from a producer who had applied the fungicide in soybeans and didn't see a change. So long and behold, when we went back and pulled samples, we found that he had resistance. So not every time you have a fungicide not working is because of resistance. It could be perhaps, the right active ingredient was not used, or maybe the fungicide was applied quite late, or maybe it wasn't mixed well. So you have to rule out other factors. Note that every time a fungicide fails is going to be due to fungicide resistance. Also, if you're suspecting fungicide resistance, please let us know. Send us a sample or call us, and we'll come and take a sample and test it in the lab. We'll subject the isolate to higher doses of the fungicide, and if the fungus is still growing in media on high concentration of a fungicide, then we know there is fungicide resistance, and then we'll do other tests following that. We also recommend, where possible, to leave untreated strip in your field when you spray fungicides, and that way you can monitor things. So if you're seeing the same level of disease where you applied a fungicide and where you didn't apply a fungicide, that would indicate pretty easily that you may have fungicide resistance issue. Also, if you leave untreated area, you can also compare when it comes to yield, whether the fungicide investment was worth it. So again, some of the guidelines for us to follow as we use fungicides to make sure that we use them sustainably for a long time. But really, the take home is, what are you treating for? Is it for white mold? Is it for frogeye? Is it for brown spot? And what is helping you to make that decision? Are you seeing enough disease pressure to warrant a fungicide, or can you not worry applying a fungicide if there's such a low disease and if the crop is already ahead in the growing season? So at this point, I can't emphasize enough the importance of accurate disease diagnosis. Sometimes there are other things that are out there, could be a bacterial disease. And if you apply a fungicide when you don't need it, not only are you losing that money, but as we saw, you could, this could lead to problems in the future, like fungicide resistance. So here again, a few reminders that field diagnosis is really important. So in case in point here, we have brown spot and we have bacterial blight, as you can see in these pictures. And these two diseases can look alike. The distinction here is that bacterial leaf blight is going to be on the top canopy, and then brown spot is mid and lower canopy. And then bacterial leaf blight, can cause the leaf to tatter or split a bit where you have this bacterial disease problem. So again, knowing how to differentiate between these different diseases is important. You can't know everything out there, so please use us. We're here to help you out as you scout and diagnose diseases. Another example here, so on the left we have frogeye leaf spot, and then here on the right we have an injury on these leaves caused by something that was applied in soybeans. So again, these things can look alike, but be careful when you're scouting so you know exactly what is going on. So accurate diagnosis and timely diagnosis is going to be really an important factor as you manage these different diseases, and not only in soybean, but any other crop. And so thinking about applying fungicide in soybeans, some of the things we have as pointers, managing white mold is a little difficult because this is a disease that you don't see symptoms. And by the time you see symptoms, it's quite late to apply a fungicide. So we recommend that where you have had white mold in the past, or where, around the soybean is starting to flower, we're having frequent rains, and you're having quick canopy closure, like where you have narrow spacing or you planted at high population. then in those conditions, you may want to think of applying a fungicide at R1. That's the best timing to control white mold. For the other diseases, like we mentioned, brown spot or Frogeye leaf spot, since these tend to develop and become more severe later in the season, you may want to delay that fungicide to around R3, R4, so that you have extended protection. The Frogeye leaf spot pathogen is favored by warmer and wet weather. So much later in the season, once we get really warm temperatures, that's when Frogeye takes off. Also when the soybeans are podding past the podding time. So the fungicide applied around R3, R4 will provide that protection for these diseases that tend to accumulate later in the growing season. So I'm going to pause here and ask Matt to show us a quick poll just for us to have an idea whether how fungicides are considered in soybeans. So if you can please respond to this poll quickly, so we kind of have an idea whether fungicides are being considered in soybeans as often as we might think. So far what I'm seeing here is that the majority of people are saying that it is not true that a fungicide would be applied in soybeans because of fungal disease pressure. And so that's pretty true with what we're seeing as we scout these fields around the state, that for the most part, we see very little disease in soybeans to require a fungicide. So I'm gonna end this polling here and then continue with the rest of the talk for the few minutes I have left. Okay, so then what I wanted to talk about also is soybean cyst nematode. So quickly here, I don't have a lot of time. So one thing we are seeing is that the soybean cyst nematode continue to spread in the state. So as of 2019, we saw one field, I believe there are actually two fields, become positive for SCN in Brewer County. So it's further extending west where soybeans grown. We believe in these other counties where we haven't found a positive field, probably it could be there. So SCN is a big problem in soybean, and we need to know how to manage this disease. Excuse me there. So quickly looking at some of the ways we can manage SCN, if we can use resistance, so we have commercial soybean varieties with SCN resistance, but as we may know, the majority of this resistance is coming from PI 88788. We have done population studies here on campus looking at the difference in the reproduction of SCN populations on the several sources of SCN resistance. So PI 88788 is still active, it's still a good source of resistance. The maximum reproduction we've seen on this line is 25%, so which means that this line can still provide protection, but over time, SCN populations are learning or have adapted and are able to reproduce on this source. So it's not really sustainable to keep using the same source. So we need to be thinking of rotating different sources of resistance. We have now a few that are coming on that have picking as a source of resistance, and also other PI lines that are being incorporated in soybean varieties. So if you have SCN and you have high numbers, I think of rotating away from PI 88788 to try and make sure that this population of nematode is not adapting and reproducing on this one source of resistance. Nematicide seed treatments, are they effective? When should we think of applying nematicide seed treatments to manage SCN? So yes and no, depending on the level of SCN in your field, then we can think of using a nematicide seed treatments. Where you have a soil test of SCN and the numbers are pretty high, more than 6,000 eggs per 100 cc of soil, then you may want to think of using nematicide seed treatment in addition to using a resistant variety. So there are a number of products, the ones that can also provide protection against the sudden death syndrome of soybean. These are two good active ingredients. We've done some testing here in the greenhouse, and they can really reduce the number of cysts that are developing on soybean roots. So for pyram and pyrethroid, those are the two active ingredients. We have other products that can help us as seed treatments to control SCN, so clavipin and avio. These two use biological control agent, a bacteria that infects the nematode and then prohibits it from reproducing on the soybeans. So one thing I could say, as I was mentioning, when we look at seed treatments, we don't see really a good bump in yield as a result of using nematicide seed treatments. But what is really clear is that when you use a resistant variety, so the first one here is a susceptible line in this table, and the second variety here is the resistant variety. But what you can see clearly is that the susceptible line is going to continue supporting SCN to reproduce in the field, whereas the resistant line here is really keeping that population down. And so that's why we use and recommend using resistant varieties, because you're keeping that population down and you're also getting 2 to 3 bushel, 3 to 4 bushel gain from using a resistant variety. So resistance actually is going to be pretty effective if used in addition to fungicide seed treatment. We can control SCN effectively. So last disease here to talk about would be Phytophthora root rot. If the computer can let me go there. So Phytophthora is one of the major diseases of soybeans, as seen here in these two pictures. Really having high number of plants that are killed by this disease. But also what can be seen is that this is an area where we're likely to have excessive moisture. And so that is a recipe for Phytophthora root rot to develop. One way to tell, of course, is to see this lesion that expands above the soil line. That will tell you that you have Phytophthora. And when it comes to management, definitely using varieties with RPS genes is going to be very, very crucial in controlling Phytophthora. One thing I wanted to mention is that on this graph, we're showing the frequency of isolates that defeated the RPS genes in a soybean variety. And these are isolates collected in the past few years. And you can see that the majority of the isolates are going to defeat the RPS 1A, 1B, 1C, and 1K, and also RPS 7. So if you're selecting a variety to control Phytophthora, you want to make sure you're using something that has these RPS genes, so 2 up to 6, or at least the combination of different resistance genes. We have seen a number of varieties now on the market that have 1K and 3A. So a variety with stacked genes would have a better chance of defeating the isolates of this pathogen, Phytophthora root rot. Seed treatments are also good, but seed treatments for Phytophthora will not last the season long. As we have seen here, these are plants dying throughout the season. So seed treatments will provide that protection. And so much later in the season, then you need resistance gene to help out. So again, don't rely only on seed treatments for managing Phytophthora. You want to select a variety with good resistance genes. The ones that, as we saw in some of the data we've collected, the RPS ones are not as effective as the upper RPS genes. So if you use seed treatments, make sure that you're using that in addition to having a good resistant variety. So those are the 3 diseases we wanted to talk about, looking at the foliar diseases, white mold, soybean cyst nematode, and then Phytophthora root rot. Because of time, I won't keep going, but if you have a question, please ask us, and we'll have an extended time for discussions, or contact us by email or phone call. So I'll pause here, and I don't know if we have a minute for Connie to add on the soybean cyst nematode, or if we can open it for questions.
Yep, Connie, go ahead if you have anything to add.
Yeah, I just wanted to make a quick mention for soybean cyst nematode that you're able to sample for that. You can sample any time of the year as long as the ground is not completely frozen or completely saturated. Ideally, the best time to sample would be after soybean harvest. With the samples, you're able to send them to the South Dakota soybean, send them to the SDSU diagnostic clinic for analysis. If you're a South Dakota soybean grower, it is free due to a grant through the South Dakota Soybean Research and Promotion Council. So we really encourage you to sample. This fall, if you haven't sampled in a while, go ahead and sample this spring, sample again in the fall to kind of check your levels. Really encourage you to kind of stay on top of it, because like we like to say, if you're seeing one cyst out there, it's one too many, and it's easier to manage when those numbers are low versus high. So again, really encourage you to send those samples into the diagnostic clinic. And with that sampling, it's only about a pint. You take about, you can do it really intensive or you can have it focused, but you would mix your cores together and send in about a pint to the diagnostic clinic. So with that, I will stop talking and open it up for that QR code.
All right, thanks, Connie. I'm going to go ahead and share my screen then so everybody can see the second QR code. And at this time, we will have Emmanuel's, the poll for Emmanuel's talk. I ask that everybody stay on for a minute because we missed mine due to a technology issue. So this poll is just to answer a couple questions regarding Emmanuel's talk. And then once we're done with Emmanuel's, we'll switch to the poll for my talk. If you need the QR code here for CCA credits, please feel free to get that now. And I'll leave that up for a couple minutes.
And Adam, as that is going on, there are a few questions in the question answer.
Yeah, you do. You have 3 questions, Emmanuel.
Okay. So the first question says, what is the percentage decrease of sclerotia when rotating away from soybean beyond no-till to minimize bringing viable sclerotia to the soil surface? Can a shallow vertical tillage be almost as effective as tilling deeper with a disc chisel? So the first part of the question, what's the percentage of decrease of sclerotia when rotating away from soybean? So actually, some of the research on the sclerotia survival has shown that if that sclerotia is left on the surface, it doesn't last more than 2 seasons. The first year, it will be attacked by other microorganisms in the environment. And as long as you have rotated away, say in wheat, in fact, we've seen this in wheat, a lot of these sclerotia that are left on the surface are going to germinate. And once they germinate and have these mushroom structures from them, then they are done. So they won't remain viable within 1 to 2 years. So that would be the period of time that sclerotia left on the surface can survive. However, if it is buried, like the second part of the question mentioned, it will remain active to remain viable for a long time. And once you do tillage again and these are brought on the surface, they will provide an inoculum for white mold. So rotating away from a white mold field, if no till is done, and so this sclerotia remain on the surface and nothing's brought in, then 1 to 2 years away from soybeans can pretty control white mold from developing. The second part, I am not sure that I have the information to answer this question. I don't know if Paul is on to help in terms of vertical tillage and tilling deeper. I'm not sure about that. But the point would be that if you do deep tillage, trying to bury this sclerotia, then you want to stay away from tilling that field again 3-4 years out, so that the sclerotia can remain deep in the soil and eventually die off. But if the following year you till again, you're going to bring the sclerotia back to the surface. I apologize if I'm not able to differentiate between vertical tillage and deep tillage. Then another question was, won't leaving a strip of untreated area possibly cause the disease to spread further? It should not, because first of all, the rest of the field will be treated, and then this is just only a small part of the field, so it shouldn't spread more than necessary. So it's not a big risk factor, especially if the rest of the field has been treated.
All right, thank you, Emmanuel, and thank you everybody for sticking around to do both polls here. So thank you for joining us all today. There will be a link to the presentation recordings in the chat. And then also, like I had mentioned earlier, look for an email from SDSU Extension on Friday that will have a survey link for feedback regarding this week's presentations. And if you have any questions about today's presentations or any concerns, please feel free to email me, or if any questions come up later, I will get those to you, Emmanuel, if they're for him or for myself.